Imagine your house gets broken into. Like a burglar smashes a window, climbs inside, steals your television, and, I don't know, leaves muddy footprints all over the rug.
Right, it's a very physical, obvious intrusion.
Exactly. And because the burglar is an independent physical intruder with their own tools, you have clear ways to fight back. You can call the cops, you can set a trap, install stronger locks, and in the world of infectious diseases, that burglar is basically a bacterium.
Yeah, it brings its own equipment to the job.
Right. But what if the intruder doesn't break a window at all? What if instead they hack your smart home's operating system from a laptop halfway around the world?
Oh, wow. Yeah, that changes the entire dynamic. Suddenly your own house is locking the doors on you, turning off the lights, commanding your own printer to print out like 10,000 copies of the hacker's manifesto.
Exactly. And that hacker is a virus. So welcome. You're joining a custom-tailored deep dive today, and our mission is to really understand how we fight these ultimate cellular hackers. Like, how do you stop a virus without shutting down the entire house?
It's basically the hardest question in pharmacology.
Right. So we are going to get into the mechanics of antiviral drugs, the massive policy blunders that cost billions of dollars, and a major misconception that most people, maybe even you listening right now, have about what happens when you take these medicines.
Yeah, it's a wild ride.
But to really grasp how we fight back, we have to look at why this was so agonizingly difficult for scientists to figure out in the first place.
Well, it really comes down to a concept called selective toxicity. Back in the early 1900s, this scientist named Paul Ehrlich came up with this idea of the magic bullet.
A magic bullet, okay.
Right, so a drug that could hunt down and kill a pathogen without significantly harming the human patient.
Yeah.
And I mean, it worked beautifully for bacteria.
Because they're like the burglars.
Exactly. We developed antibiotics because bacteria have their own independent cellular machinery. Like they have cell walls, which human cells don't have. They have their own unique ribosomes for making proteins.
So you just design a drug that poisons those specific bacterial parts. Easy enough.
Right, you target the things we don't share. But a virus doesn't bring its own factory. It travels incredibly light. It's just a tiny envelope of genetic code.
It's just a flash drive.
Literally. It hijacks the human host's ribosomes, our energy supply, our amino acids. There's no independent cell wall to poison.
Which means if you tried to just poison the virus, you are, by definition, poisoning the human.
Exactly. To make an antiviral, you have to find an incredibly subtle biochemical difference, like a specific viral enzyme or a unique step in their takeover process that has absolutely no human counterpart.
And our first attempts to do this were, honestly, basically disasters. Like, we tried the sledgehammer approach first.
Yeah, it was not pretty.
In 1963, a Yale researcher named William Prusoff created the first clinically approved antiviral called idoxuridine. It was designed for this really nasty herpes infection in the eye.
Right.
And the drug was basically a fake DNA building block meant to jam up the virus's ability to copy its genetic code, but it didn't discriminate.
No, it lacked that true selective toxicity. I mean, it jammed up human DNA synthesis just as effectively as the viral DNA synthesis.
Oh, man.
The drug was so brutally toxic to human cells that it could only be applied as topical eye drops. Like if you put it in someone's bloodstream, the collateral damage to their own tissues would be catastrophic.
Okay, let's unpack this. How did we get from toxic eye drops to pills you can take every day for years? Because clearly we don't use the sledgehammer anymore.
No, thankfully. The paradigm shifted completely in the late 1970s, and that was thanks to two researchers at Burroughs, Wellcome Gertrude Eline, and Howard Schaefer.
Right.
They pioneered this concept called rational drug design. So instead of just throwing thousands of random chemicals at infected cells in a petri dish and hoping one worked without killing the cell.
Which is basically what they were doing before.
Exactly. Instead of that, they started looking for specific exploitable weaknesses in the virus's chemistry. And this work was so revolutionary it eventually won a Nobel Prize.
They created a cyclover, right? Which is still used constantly today for herpes viruses. But what made a cyclover so different from the sledgehammer?
Well, it's essentially a brilliant biochemical trap. A cyclover is also a fake DNA building block. It's a lookalike for a natural molecule called guanosine. But Schaefer and Eline designed it so that it stays completely inert inside a normal, uninfected human cell.
It just floats around.
Yeah, it just floats around doing absolutely nothing.
I have to push back on that, though, because it's a chemical compound. It's not a smart bomb. It doesn't have a brain. how does a cyclover know it's inside an infected cell versus a healthy one?
It doesn't know. It waits to be activated.
Uh,
So for this fake building block to be used in constructing a DNA chain, it needs a chemical tag-a-phosphate group attached to it.
okay.
And human cellular enzymes are terrible at attaching that first phosphate group to a cyclover. They mostly just ignore it. But the herpes virus brings along its own unique tool. It's an enzyme called thymidine kinase.
Oh, I see where this is going.
Right. And that viral enzyme happens to be roughly 100 times more efficient at grabbing a cyclover and slapping that first phosphate onto it.
Oh, that is incredibly clever.
Yeah.
So the drug requires the hacker's own specific tool to get armed.
Precisely. The virus essentially activates the weapon that kills it. Once the viral enzyme attaches that first tag, the human enzymes finish the job, and the drug gets pulled into the growing chain of viral DNA.
But it's a fake piece.
Exactly, because a cyclover is chemically incomplete. It's missing the hook needed to attach the next piece of DNA. So the whole assembly line just crashes.
Wow.
The DNA chain terminates. The virus can't replicate. And because this activation almost entirely happens in cells where the viral enzyme is present, a patient can take a cyclover orally with very mild side effects.
We trick the virus into printing a corrupted string of code that crashes its own server. That is brilliant. So if we can shut down the hacker this effectively, does this mean the patient is completely cured?
Oh.
Because, like, if I take antibiotics for strep throat, the bacteria die, they get cleared out, and I am cured. Is it the same here?
See, this raises an important question, and it's probably the single most misunderstood aspect of virology.
Really?
Yeah. The short answer is no. Over the last 40 years, the medical definition of success for antivirals had to shift drastically. We moved away from the idea of a cure, meaning, sterilizing the body and eliminating every trace of the pathogen. Instead, the goal became being virus static.
Virus static, meaning static, like keeping it in one place, just suppressing it.
Yes, exactly. Most modern antivirals drive viral replication down to incredibly low, often undetectable levels. They hold the line, which gives your immune system the upper hand.
But they don't get rid of it entirely.
No, they do not clear every single viral genome from your body. So with a cyclover, the herpes virus just retreats into your nerve bundles and goes dormant.
Right.
And with HIV antiretroviral therapy, we can reduce the viral load in the blood to zero, meaning the patient is healthy and cannot transmit the virus. But latent pieces of HIV DNA remain permanently stitched into the patient's resting immune cells.
Wow. So to go back to our house analogy, we aren't actually kicking the hacker out of the house. We're just locking them in the basement, taking away their laptop, and hoping they never find another one.
That perfectly captures the reality for millions of patients. The hacker is still in the basement.
So if we know we are just suppressing them, how exactly do we confiscate that laptop? Because viruses don't just have one trick, right? They have a very specific hostile takeover process.
They do. And virologists break this takeover into a 6-stage assembly line. Literally every antiviral drug we have targets a vulnerability in one of these 6 stages.
Okay, walk us through them.
Sure. So stage 1 is attachment and entry. That's where the virus finds a specific receptor on your cell and forces its way in.
Got it.
Stage 2 is uncoating. It sheds its protective protein shell to release its genetic payload into your cellular machinery.
Right, so the hacker is inside and unpacking their zip files.
Exactly. Stage 3 is genome replication. That's forcing the cell to make thousands of copies of the virus's raw genetic code.
So that's what a cyclover attacks.
Right. Then stage 4 is transcription and translation. That's hijacking the cell's ribosomes to manufacture the physical viral proteins needed to build new viruses.
Building the physical parts.
Yep. Stage 5 is assembly, snapping those new genomes and proteins together into functional units. And finally, stage 6 is release, where the new army of viruses breaks out of the cell to infect the neighborhood.
It's a literal factory floor.
Right.
And so you mentioned a cyclover attacks stage 3, the genome replication, but relying on this one trick, fooling the virus with a fake building block, that has a massive vulnerability, right? Because viruses don't just sit there and let you poison them, they evolve.
They evolve constantly.
And the story of HIV drugs is the ultimate cautionary tale of what happens when that assembly line fights back.
It truly is. I mean, the very first drug approved for HIV was AZT. And what's fascinating here is that AZT wasn't even invented for HIV.
Really?
No, it was synthesized in 1964 as a potential cancer drug by a researcher named Jerome Horwitz, but it failed and literally sat on a shelf for 2 decades. Yeah, and then in 1985, as the AIDS epidemic was exploding, researchers at the National Cancer Institute pulled it off the shelf and tested it against HIV.
And the FDA fast-tracked it in record time. I think it was just 25 months to approval in 1987. And AZT also targeted stage 3. It was another fake building block meant to terminate the HIV DNA chain. But as a long-term solution, it was a failure.
It bought patients a little bit of time, yes, but HIV is an RNA virus that makes millions of copies of itself every day, and it makes a lot of mistakes when it copies.
Meaning it mutates incredibly fast.
Very fast. And because patients were only taking AZT-like, a monotherapy, it only took a few months for the virus to stumble upon a mutation that allowed its enzymes to recognize AZT as a fake and simply reject it.
It just spat it out.
Yeah, the virus bypassed the drug entirely.
So what does this all mean for the patient? If you target a specific stage and the virus just mutates around the drug, aren't we back at square one? Like if you throw 3 different drugs at it instead of 1, wouldn't you just create a super resistant virus 3 times as fast?
Well, if we connect this to the bigger picture, you have to look at the sheer mathematical probability of evolution. This is what David Ho realized in the mid-1990s.
Okay.
He championed a strategy called hit early, hit hard. Let's say a virus has a 1 in 10,000 depressed chance of developing a mutation to escape drug A. If you only use drug A, it's going to hit that lottery eventually.
Right.
But what if you give the patient drug A, drug B, and drug C all at exactly the same time,
Yeah.
and they all attack different stages of the assembly line?
Ah, I see. You changed the math.
Radically.
Yeah.
We introduced protease inhibitors, which attack stage 4.
And what does a protease inhibitor actually do? Because that sounds like dense medical jargon.
Right, sorry. Think of it like a manufacturing defect.
Yeah.
When HIV forces your cell to make viral proteins, it doesn't make them one by one. It prints out one massive long string of protein, like a solid block of plastic. To be useful, that block has to be cut into specific functional pieces by an enzyme called a protease. It basically acts like chemical scissors.
So a protease inhibitor.
It gums up those scissors. So the virus ends up with useless, uncut blocks of plastic instead of functional parts.
So in this hit-hard strategy, you're jamming up the genome, copying at stage 3, and simultaneously gluing the scissors shut at stage 4.
Yes, and this became known as highly active antiretroviral therapy. For the virus to survive, it doesn't just need one lucky mutation. It needs to develop 3 independent, highly specific mutations at the exact same moment.
Which is almost impossible.
The odds are infinitesimal. It's like trying to win the Powerball 3 times on the same Tuesday.
That's a great analogy. And the real-world impact of that mathematical shift was staggering. Between 1996 and 1997, after this combination therapy was introduced, AIDS deaths in the United States plummeted by roughly 40 to 50%.
Incredible.
It is still considered one of the most dramatic reversals of a chronic infectious disease in modern medical history.
It absolutely proved that if you attack multiple stages of the assembly line at once, you can outsmart the hacker.
But knowing where to hit the virus is only half the battle. Because recent history with the COVID-19 pandemic taught us a brutal lesson about when you strike. Timing, it turns out, dictates whether a drug is a miracle or a complete dud.
Yeah, timing is everything in virology. We saw this play out vividly between 2 of the most famous COVID-19 drugs, remdesivir and Paxlovid.
Let's start with remdesivir. It was one of the first antivirals authorized for COVID back in 2020, and it targets stage 3, just like a cyclover or AZT. It tries to wreck the genome replication.
Right.
But the World Health Organization ran a massive global study, the Solidarity trial, looking at over 11,000 hospitalized patients. And the results were incredibly underwhelming. Remdesivir had little to no effect on overall mortality.
Yeah, it showed some modest ability to shorten recovery times for specific groups, but as a life-saving tool for severe patients, it totally fell flat.
But then there's Paxlovid.
Exactly. Now look at Paxlovid. It's a combination drug, and its main component is a stage 4 protease inhibitor.
Gluing the scissors shut.
Yes, preventing it from cutting up its raw proteins. And in clinical trials, when Paxlovid was given to high-risk patients within the first few days of symptoms, it reduced the risk of hospitalization or death by nearly 89%.
That is a massive difference. So why did one fail to save lives and the other succeed so dramatically? Was remdesivir just a bad drug?
It wasn't necessarily a bad drug. It was used at the wrong time in the disease's life cycle. Remdesivir was largely tested and used on patients who were already hospitalized.
Right.
And by the time someone is sick enough to need a hospital for a respiratory virus like COVID-19, the peak window of viral replication is often already passed.
The virus has already finished its major manufacturing run.
Yes. The severe damage happening in the lungs at that late stage isn't mostly being caused by the virus replicating anymore. It's being caused by the patient's own immune system overreacting and destroying tissue in a hyperinflammatory response.
Oh, I see.
So giving an antiviral at that point is basically shutting down an assembly line that is already closed for the day.
Okay, so it's like putting out a fire. Using Paxlovid early, like within the first 3 days, is like blasting your kitchen with a fire extinguisher the second the toaster sparks. You stop the spread immediately.
Exactly.
But using remdesivir on a patient who is already in the ICU on oxygen, that's like spraying the kitchen with a fire extinguisher after the fire has already spread to the walls and the roof is collapsing. Extinguishing the toaster doesn't help when the structural damage to the house is already done.
That is a very precise way to look at the timeline of viral pathology. Paxlovid exploits that narrow, critical window before the cascade of immune damage begins.
Here's where it gets really interesting, though, because there is a third COVID drug that took a completely radical philosophical approach. Instead of jamming up the genome copying or gluing the protein scissors shut, this drug basically says, go ahead and copy yourself, but here is a corrupted blueprint.
Yeah, you're talking about molnupiravir.
Right.
This drug utilized a really controversial strategy called lethal mutagenesis. It doesn't actually try to stop the virus from replicating. It gets absorbed by the virus, and as the virus copies its RNA, the drug forces the copying enzyme to make mistakes over and over again.
It intentionally scrambles the code.
Heavily. The goal is to induce such an incredibly high rate of genetic errors that the resulting new viruses are completely defective. They are so mutated they can't function. The virus literally mutates itself into extinction.
Well, hold on. If we are intentionally encouraging a virus to mutate, isn't that playing with fire? What if it mutates into something worse?
Yeah, that exact concern sparked a major scientific debate. Virologists raised two massive red flags. First, what if the mutagenesis is sublethal?
Meaning it doesn't kill it.
Right. What if the drug causes mutations, but the patient doesn't take the full course or their immune system is weak and the virus actually survives those mutations?
Yeah.
Could we theoretically be helping the virus invent new, dangerous variants?
Just speeding up its evolution for it. That's terrifying.
Exactly. And the second concern was about the host. If a drug is this good at scrambling RNA, could it accidentally get incorporated into human DNA and cause mutations in the patient's own cells?
Oh, wow.
Because of these very serious theoretical risks, molnupiravir was heavily restricted. It was not authorized for pregnant patients, and it became a second-line option compared to Paxlovid.
It's a stark reminder that we are dealing with immense biological power here. Like, we can interrupt or corrupt these hackers, but if we don't fully understand the tools we're using, it can completely backfire.
Absolutely.
Which transitions us perfectly into the final phase of this deep dive, the massive pitfalls and public policy blunders surrounding antivirals. Because misunderstanding these drugs hasn't just sparked academic debates, it has caused billion-dollar public health crises.
It really has.
Let's look at the most common misconception, the one doctors hear every single winter.
Oh, I know exactly what you're going to say.
Yeah.
I have a terrible cold or a flu, so I need an antibiotic to feel better.
Yes. Why is that so fundamentally wrong?
Because of the selective toxicity we talked about at the very beginning, antibiotics are precision engineered to attack bacterial structures, like destroying a bacterial cell wall or gumming up bacterial ribosomes.
But viruses don't have those.
Right. Viruses do not have cell walls. They do not have their own ribosomes. So taking an antibiotic for a viral cold is like trying to unlock your front door with a car key. The mechanism simply does not apply.
It does nothing.
Not only does it do nothing to the virus, but it actively harms the patient's microbiome and fuels global antibiotic resistance.
Okay, here's another myth. If an antiviral works on one virus, it should work on most of them. It's like a broad-spectrum shield.
Also incredibly false. The enzymes these drugs target are hyperspecific lock-and key mechanisms. Even viruses within the same family can have structural differences in their enzymes that render a drug useless.
So you can't just mix and match.
No, you can't just take an HIV protease inhibitor and expect it to work on the COVID-19 protease. Their 3D shapes are different. That's why developing broad-spectrum antivirals, like a drug that works against many different virus families, is one of the absolute hardest challenges in modern pharmacology.
Let's talk about the big one, though, the billion-dollar mistake. The idea that Tamiflu is a cure-all for influenza.
What's fascinating here is how a subtle misunderstanding of data scaled up to national policy. Oseltamivir, known commercially as Tamiflu, is a stage 6 inhibitor.
Release stage.
Right. It doesn't stop the flu virus from replicating. It stops the newly built viruses from budding off and exiting the infected cell. And in the 2000s, fearing a devastating avian flu pandemic, governments around the world panicked. They believed Tamiflu would significantly reduce hospitalizations and severe complications, so they stockpiled it.
The numbers on this are staggering. The UK government spent roughly £424 million, and the United States spent over $1 billion filling warehouses with this drug.
Yeah, but the problem was, the major claims about preventing severe complications were based on clinical trial data that was largely unpublished or only partially released by the drug's manufacturer, Roche.
Which is a huge red flag.
Independent researchers couldn't actually verify the boldest claims.
Right, until a group called the Cochrane Collaboration stepped in.
Yes, they're an independent network of researchers led by epidemiologist Tom Jefferson, and they spent years fighting to get the raw, unpublished clinical study reports. It's a massive transparency battle.
And when they finally analyzed the complete data in 2014, the findings were incredibly sobering.
Very.
They found out the billion-dollar stockpile was basically for a mild symptom reliever, right?
Essentially, yeah. The data show that Tamiflu did not definitively reduce the risk of hospitalizations or serious complications like pneumonia.
So what did it do?
What it did do was shorten the duration of flu symptoms by roughly half a day to 1 full day. And even then, it only worked if the patient took it within 48 hours of feeling sick.
So governments hoarded billions of dollars worth of a drug to prepare for a catastrophic pandemic, only to find out it was essentially a fire extinguisher that barely dampens the flames, and only if you manage to spray it in the first 10 seconds.
It was a massive wake-up call for public health policy. It proved that you cannot base national stockpiling decisions on incomplete or secretive trial data. It actually helped kickstart the all trials movement, demanding that every single clinical trial be registered and its results reported publicly.
But even when we have an influenza drug that actually does work, we still manage to mess it up. Like, I want to talk about amantadine. Because this drug stops the flu virus at stage 2, it prevents it from uncoding and unpacking its genetic material. But doctors don't really prescribe it for the flu anymore, do they? And the reason we lost it is shocking.
Yeah, amantadine is a textbook tragedy of evolutionary pressure and overuse. It was highly effective, and it was used widely for human treatment, but it was cheap to produce.
Oh, no.
So in several countries, massive agricultural operations started using it not for humans, but for poultry.
They were dumping human antivirals into chicken feed.
Yes, to protect massive commercial flocks from avian flu outbreaks.
Yeah.
Now think about the scale of that. Millions upon millions of birds housed in tight quarters, all taking a prophylactic antiviral.
It's basically an evolution factory.
You are creating the ultimate evolutionary training ground for the virus. The virus mutated rapidly to survive the drug. And because flu strains jump between birds and humans, those highly resistant mutated strains spread globally.
We literally gave the hacker the blueprints to our own firewall so we could save some chickens.
We did. The resistance rate spiked so high that by 2006, the CDC had to issue a blanket recommendation to stop using amantadine and its cousin rimantadine for influenza in the United States.
Wow.
Through sheer carelessness and lack of global regulation, we burned through the usefulness of an entire class of drugs in just a few decades.
It shows you how relentless this biological arms race really is. But before we wrap up this deep dive, I do want to highlight the one glaring, miraculous exception to almost everything we've discussed today. Because we said at the beginning that antivirals are virostatic. They suppress, they manage, but they don't cure.
Right.
However, there is one major disease where we actually did clear the house: hepatitis C.
Hepatitis C is the shining beacon of hope in this field. For decades, a Hep C diagnosis meant a slow, agonizing march toward liver cirrhosis or liver cancer with treatments that were highly toxic and barely worked.
But then things changed.
Dramatically. In the 2010s, researchers developed direct-acting antivirals, or DAAs, drugs like sofosbuvir.
And these weren't just monotherapies, right? They used the same logic as the HIV treatments.
Exactly. DAAs combine multiple agents that attack different specific viral enzymes simultaneously. They hit hard and hit fast. But unlike HIV, which hides its DNA inside our cells, hepatitis C doesn't integrate into the human genome in the same way.
Which is the key difference.
Yes. Because of that, this multi-drug assault doesn't just suppress the virus, it completely eradicates it.
A functional cure.
Over a 95% cure rate. You take a course of pills for 8 to 12 weeks with minimal side effects, and a chronic, deadly viral infection is simply gone. It is one of the greatest scientific triumphs of our generation.
Though it is definitely worth noting that this medical miracle kicked off an entirely different kind of policy war. When Gilead Sciences first priced this cure, a 12-week course cost around $84,000.
Yeah, that was highly controversial.
It sparked an immense ethical and economic debate about whether a drug that cures a public health crisis should be priced based on the cost of innovation and long-term healthcare savings, or priced to ensure immediate, universal access. It basically proves that even when the science is flawless, the application is always messy.
That tension is always there.
Yeah.
But biologically, DAAs prove that a true cure is possible if we can understand the viral machinery deeply enough.
So, what does this all mean for us? To bring it all together. Antivirals are not broad-spectrum sledgehammers. They are incredibly precise tools designed to exploit selective toxicity targeting the hacker's assembly line without destroying the human factory.
Exactly.
We've learned that to beat a rapidly mutating enemy, you have to hit early, and you have to hit hard with combined forces. And perhaps most importantly, we learned that for the vast majority of viral infections, these drugs are virus-static. They don't cure us, they just keep the enemy in check.
They hold the line. Without these drugs, HIV would still be a universal death sentence instead of a manageable condition. They dictate our pandemic readiness, and they shape the future of modern medicine.
They absolutely do. But I want to leave you with a final thought to mull over. If it's true that most antivirals don't actually clear the infection, but just suppress the virus to undetectable levels, that means millions of people are walking around right now with dormant viral DNA permanently integrated into their cells.
It's a lot of people.
The herpes virus sleeping in the nerves, HIV resting in the immune cells. Our bodies are basically living museums of suppressed viruses. How might these permanent, silent viral hitchhikers be subtly shaping human biology, altering our long-term health, or even driving the future evolution of our immune systems in ways we haven't even begun to measure? If our smart home still has the hacker's dormant code hiding deep in the basement server, what happens when that code finally decides to wake up? Thank you so much for joining us on this deep dive. Keep asking questions, and we will see you next time.